Hello everyone, you are welcome to C Academy YouTube channel. My name is Veleleni Nkosi. In this lesson we discuss the structure and the functions of an RNA. By the end of this lesson we will be able to differentiate RNA and the DNA molecules. Without any further ado, let's get to it. So what you see on your screen is a lesson structure of today. This lesson structure is based on the frequent asked questions about RNA. So these are the questions that appear most of the time during examination. So I will read the questions so that you'll be able to know what to expect in this lesson. So number 1, we define an RNA molecule. Then number 2, we describe the locations of an RNA in a cell. Then number 3, we describe the structure of an RNA. Then number 4, we'll know what is the role that the RNA plays in a cell. Then the last one, we will compare the DNA and the RNA. So this is the lesson, the structure of this lesson. So I hope you will enjoy. So let's start with number 1. Number 1, define an RNA molecule. So RNA or ribonucleic acid is a single-stranded linear polymer composed of nucleotide. If you remember, we learned about DNA. So DNA is double-stranded. So RNA is single-stranded. Both DNA and RNA, they are polymer structure. So that means they are built from a monomer structure. So that monomer structure is a nucleotide. So the definition of a nucleotide is a monomer or a building block that comes together to create DNA and RNA. Like I say, if you pack nucleotide together, you ended up with DNA or RNA. So nucleotide is like a brick, and then DNA and RNA is like a house. So we use bricks to build a house. So and then a nucleotide is made up of 3 key components. The components are phosphate group, sugar molecules, and the nitrogenous bases. So this structure here is a simple demonstration of a nucleotide. We've got P here, which is the phosphate group, and then you have S here, which is the sugar molecules, and then NB, which is the nitrogenous base. And this nitrogenous base, we have 4 types of nitrogenous base. We have adenine, we have uracil, we have guanine, and we have cytosine. So each nucleotide contains one of these nitrogenous bases. So this is the definition of an RNA. You must know this is the basic structure. It's built from the nucleotides. And then going back and to our lesson structure, describe the locations of an RNA in a cell. So we have 3 types of RNAs. We have messenger RNA, which is mRNA. mRNA is formed in the nucleus, but its functions in the cytoplasm. This is the simple structure of an mRNA. It's formed in a nucleus, but it functions in a cytoplasm. So we need to know that 3 adjacent nucleotides of a mRNA are called codons. 3 adjacent nucleotides are called codons. So this is very important. You must know that it formed in a nucleus and then functions in a cytoplasm. And then another RNA, we have ribosomal RNA, which is rRNA. This one is found in the ribosome in the cytoplasm of the cell. So this is a ribosome. Inside this ribosome there is rRNA. rRNA together with proteins, they build ribosome. So we have another type of RNA inside the ribosome. And another RNA is a transfer RNA. which is tRNA. tRNA is found freely in the cytoplasm of the cell. So this is the simple demonstration of a tRNA. It contains amino acid and then it has anticodons. Here we said codons, but this one, it has anticodons. So I will show you the relationship between anticodons and the codons when I explain the role of a tRNA. So you must know these locations of which RNA is found in which part of the cell. So let's go. Describe the structure of an RNA. So describing the structure of an RNA, here is the structure. RNA is single-stranded, not a coil. So we know that is single strand, and then it consists of a monomer. The monomers is called nucleotides, which link to form a longer chain. So I've talked about it, that a nucleotide is like a brick, and then RNA is like it's a house. We use bricks to build a house. And then the sugar in RNA is called ribose. So the sugar of an RNA is called ribose, while the sugar of a DNA is called deoxyribose. So that is another different. And then the nitrogenous bases of an RNA are adenine, uracil, cytosine, and guanine. So here we've got something, uracil. In DNA we had thymine. So to show you the structure here, this is the structure of an RNA. You must be able to label this structure, or you must be able to draw the structure and label. So like they say, it's a single strand unwind. It's single strand and it's not winded. It's just like straight. And then it's built of nucleotides. So let me show you one nucleotide. This is one nucleotide. This one. We've got phosphate, we've got sugar molecule, and we've got nitrogenous base. This nitrogenous base is guanine. So, and then we've got another nitrogenous base here, we've got another one here. And then we must know that RNA doesn't have thymine. We have uracil. This U represent uracil instead of thymine in DNA. So, but in RNA we have uracil. So that is another different. And the sugar that is in RNA is called ribose sugar. So it's not deoxyribose sugar. So it's called ribose sugar. This is the structure of an RNA. So we must be able to interpret this structure. Note that phosphate is bound to sugar molecule, and then nitrogenous base is also bound to sugar molecule. So this is the structure that they're talking about. You must be able to draw, and then you must be able to explain it. You must be able to describe it, because sometimes they will tell you to describe the structure of an RNA. So go into our lesson structure. What is the role that RNA plays in a cell? So the role of an RNA, this is the function of an RNA. The most important role of an RNA is the transcription and the delivery of a genetic instruction from the nucleus to the cytoplasm where proteins are made. So this is the function of an RNA. mRNA will take the information from the DNA in the nucleus, then take it out of the nucleus to the cytoplasm, so that protein are made in the cytoplasm. So here's the process. mRNA copy the genetic instruction from the DNA in the nucleus. Then mRNA exit the nucleus, travel to the cytoplasm, attaching to a ribosome. And then tRNA located in the cytoplasm, carrying amino acids to form polypeptide chain in the ribosome. So to show you the process, here we have a nucleus. In the nucleus, we know that there is DNA. This is the DNA, and then So this is tRNA. It's a transfer RNA. This transfer RNA, it has an anticodon. These three nucleotides are called anticodons. So what happened is mRNA will copy this information of the DNA. This is the information from the DNA. After copying this information, will exit this nucleus via this pore. This is a nucleopore. Then will exit this nucleus, move out of the nucleus, then attach itself on the ribosome. Then the ribosome will read the codons. This is anticodons, this is codons. So what happens is the ribosome will match anticodons with codons. So this ribosome will read this nucleotide. Here we have C, we have A, C, C, which is adenine, cytosine, cytosine. And then we know that adenine in RNA bond with uracil, while cytosine bond with guanine, and then cytosine bond with guanine. So we need tRNA that has anticodon of uracil, guanine, guanine, which is this one, which is uracil, guanine, guanine. So this one will bond here. Then with the help of the ribosome, this tRNA, it has amino acids here, carries amino acid. Let me draw. This is amino acids. Let me write amino. So we've got amino acid. This is amino acid, and then what happens is this ribosome will read the mRNA again. So here we've got another codon, which is UAG. And then we know U pairs with A, while A pairs with U, and then G pairs with C. So here we need AUC. So this is the tRNA that will fit here. And then this tRNA will also bond here, which is A, U, and G, C. And then again, this tRNA, it has amino acid here, carries amino acid. After the tRNA has passed with mRNA, these amino acids, they will bond. There is a bond. Then after bonding, the first tRNA will leave this ribosome, move out and go and look for another amino acid. This one will go, and then another tRNA will come here, then join here, which is CUA. The following one will be GAU. So we'll bond here. It will also leave amino acids here, and then these amino acids will bond, and then this one will also leave. This tRNA will leave the process up until we have a lot of amino acids here. Then it takes about more than 50 amino acids to make a protein. If they are not yet made the protein, we call them polypeptide. These amino acids, when they are bound together, if they are not yet made a protein, we call them polypeptide. And the bond, this bond in between, this bond here, let me change the color, let me make something here. This bond is called peptide bond. This thing is called polypeptide, but if it has already made a protein, we will call it a protein. So if it's not yet made a protein, then it's a polypeptide. So that is the process of making a protein. mRNA will read this information here and move out of the nucleus and link itself on a ribosome, and then these tRNAs, they will move and then join, matching their anticodon with codon. Then this bond is made. This thing is happened inside the ribosome. This ribosome, like I make it small, but it's supposed to be big. These proteins must be inside the ribosome. So this is the role of a mRNA. So next we go to the comparison between DNA and the RNA. So now we will compare the DNA and the RNA. So structure, DNA, double-stranded helix, RNA, single-stranded. So structurally, one is double-stranded, one is single-stranded. The sugar in DNA, the sugar is deoxyribose. In RNA, sugar is ribose. And then nitrogenous base. DNA has adenine, thymine, guanine, cytosine. But RNA has adenine, uracil, guanine, cytosine. So DNA do not have uracil, while RNA do not have thymine. So that is another comparison and the different locations. The location DNA is found in the nucleus, and then RNA is found in a cytoplasm and the nucleus. So like they say, it's formed in the nucleus but made its function in the cytoplasm. And then the functions of a DNA is to store genetic information. And then the functions of an RNA carries genetic information from the DNA and translate it to the protein. This is the function of a RNA. It carries the genetic information from the DNA and then translate it into the protein. That is the process that I just explained of making a protein. So that is all for today, for this lesson. I hope you enjoy. Anyone who is studying, I say good luck with your studies. Thank you.